Our next speaker is Dr. Ennio Tasciotti. Sorry?
Tasciotti.
I'm sorry.
No,
And Dr. Tasciotti had his degree from the normal Scuola Normale di Pisa, and then the International School for Advanced Studies in Trieste. He has a Master of Science degree in biochemistry, Bachelor of Science in molecular biology, and a PhD in molecular medicine. He's currently an assistant professor in the Division of Nanomedicine at the Institute for Molecular Medicine at the Health Science Center in Houston. And he's going to talk to us about nanoporous silicon for drug delivery and for the discovery of the proteomic signatures for cancer detection. And again, as are several of our speakers, is going to address the notion of a personalized medicine.
Thank you.
Thank you.
Well, I don't know really how to thank again Temple and Genesis. Everybody did it in a different way. I want to do it in Italian. Grazie veramente. Siete state fantastiche. I really didn't know how to say that in a different way. Now, I really couldn't decide what to talk about. If I wanted to talk about these personalized intervention and personalized medicine after the talk that I heard yesterday, but I also wanted to say something about these other technology that we developed, because I think it's very close to the nanobots that I've heard about yesterday. And so I decided, okay, I'm going to say a little bit of both. I hope I'll be clear enough to say everything. Now, it's pretty clear for everybody in the audience that tumor needs a vascular supply and blood supply to grow, and this is somehow a bad thing because also they use the same vessels to spread the metastatic cells around. But it might also be a good way to retrieve information from the tumor, and this is true not only on a cartoon, but it's also true on a real animal. So these are papillomas that are growing on an animal, and you can see if you flip the skin that there is like a huge, massive formation of vessels. And the reason why we want to look at what's inside the vessel is that basically today everybody can image a tumor when it's already in the symptomatic phase. A good pathologist can read a slide and tell if the biopsy is positive. But the problem is that when you're in this asymptomatic phase, there are very few assays at the moment that can tell you something. And that's a very important stage of the tumor in which the metastatic cells have not been spread yet. And if you get this tumor at this stage, you might have better chances to kill it and get rid of it. Now, the reason why we wanted to look at proteomic signatures rather than genetic and genomic signatures, as many others did, is that basically DNA carries all the information, but at the end of the day, proteins do the work, and we're made of proteins. So if you look at these two animals, they have the same genes, exactly the same genes, but proteins are quite different, and the animal end up being very different. So proteins are actually the real biomarkers that we should look at. There is one big problem, though. I mean, the most readily available source of information in the body is blood. It's a very easy biological sample to get. Problem is it's very complex. Nobody really knows how many different type of proteins. There might be even millions of different type of proteins, most of which are not very interesting because more than 95% of these proteins are highly abundant. They're common among all of us. There's no information in them. So we really have to find a way to take these 95% away and focus on the residual 5%. That's not very easy. Now, that's the approach that we proposed. My boss, I mean, he has a long career in porous silicon material. And when I joined the lab 3 years ago, I learned that they knew how to make surfaces like chips with nanopores. And another thing that they're very good at doing is that they can tailor the size of these nanopores. So they can be 2 nanometers, 2.5, 3, down to 20 nanometers. And they're very irregular in structure. You see they're very parallel. They're very uniform. And I mean, it's pretty obvious the use that we want to do with them. Basically, we have these chips with these pores. We spot on them the very complex solution like serum, plasma, or blood. Of course, the bigger protein can't make it inside the pores. The smaller can. You wash away what's on the surface. You recover through some standard elution procedures what's inside. It's a very easy, straightforward. Even my grandmother got it when I explained it to her. So I think it's something that can work. And then you analyze what's in the elution. Now, just to show that that is something true, I don't know how familiar you are with MALDI scans and MALDI spectra, but basically you have to imagine that each one of these peaks is a different protein. Now, this is all the serum. If you take it from a patient and you spot it on the MALDI plate, very few peaks. Now, with our system, after the washings and elutions, if you go back and look at it, you see that during the washings, you progressively eliminate all these proteins. And the interesting part is that when you analyze the elution down in the last panel, you have a larger number of peaks, and that is exactly what we wanted to get. That we want to mine the information that is inside that fraction of the human circulating proteome. As I said, I mean, the tools that I had in the lab were this whole set of different chips with different size, different pore size. And what we built like somehow like a molecular cutoff rule. So we decided, we put together some few recombinant proteins. We mixed them. They were different in size and different surface charges. And of course, we established what was the molecular cutoff that allowed what was the pore size and what was the molecular cutoff associated with it. We also changed the structure of the pores, and also the structure of the pores has a role in which type of proteins are captured. And these are the different MALDI scans. I'm just want to go quick. Of course, you really retrieve a lot of information with this type of approach. And of course, a good bioinformatic tool and a good tool to manage all the information is something that you really need. And in this, in this slide, you see that how we somehow try to organize this data. Basically, here you have 3 different organizational panel in which we have divided the proteins that we picked by pore size, by hydrophobicity, that is the surface charge and the chemical modification of the chips that we use, or by the swelling agent that gives somehow the structure of the pores. So if you have a cubic, hexagonal, and you see that there is a differential. When you see red, it means that you have a high signal. When you see green, you have a low signal. And basically, according with the different type of surface modification, pore size, or pore structure, you have a differential signal that is retrieved from these chips. This means that these chips, without having any beta, without having any affinity, they can anyways selectively enrich a different fraction of the proteome. And this is very important when you're doing some type of biomarker discovery and you really don't know what you're looking for. So it's very important to have something that is just label-free, completely multiplexed, and let's say blind. And this is what I wanted to give as a final information. Now, if you check the amount of proteins that you can distinguish in a drop of blood, and we're talking about 10 microliters of serum, so it's a very small sample, you can detect almost 100 different peaks. Now, with our chips, and we have used only 5 different chips, we could pick up 365 final proteins. And just for the sake of time, I just put 5, but we can build like combinatorial chips with thousands, theoretically, thousands of different chips. So the amount of information that we can recover, it's really massive. And you see the difference from the 100 peaks here and the higher amount of peaks that you have in the panel below. Now, another very important thing is that we discovered while we were developing this technology is that once we spot the serum or the blood or the plasma on these chips, there is no protein degradation whatsoever. And the reason is that all the proteases that are contained in blood are bigger than 5 nanometers. So if you keep the size of your pores below 5 nanometers, you collect all the peptides, but there is no proteases that can get inside the pores and create all those artifacts and false negatives or false positives that are very often found with other technology. And so this is a very powerful thing, because now you can store a biological sample for 1 month at room temperature, no need for refrigerators, no need for freezers, and the sample is just fine when you recover it after 1 month. And this is very powerful. We like that feature for developing countries and for places that don't get our same treatments in the medical care system. And this is a little bit some insight on how we described the inhibition of protease degradation. As I said before, the system works with serum, with plasma, even with blood, and this is very convenient because you can just poke a hole in your thumb and just drop a blood on the chip, and the chip will anyways do its job. It will retrieve the information. You can just keep the chip on your bench, and after 1 month you go back and you wash the surface and you retrieve the proteins. The proteins are not degraded and are exactly as they would have been if you had done the procedure right away. And this is the reason why we have a great reproducibility. It's something that you want to do if you want to use this type of approach in a medical setting. And also, the thing that we're doing now is we're implementing this strategy for a high throughput processing. As I said before, we can have up to 1,000 different chips, so 1,000 different combinations of surface charge, pore structure, and pore size. And given the fact that we only need 10 microliters of serum, it's really feasible to analyze a lot of different chips, but it takes time, and you want to do it in a very standardized way. And so we developed these masks. We developed this mask, and there are different type of chips, different chemistry and everything, and every one of these work with an automatic robot, and this save a lot of time and makes it very convenient. Now, this is the last panel that I put together, because that's what we envision to do with this technology. Now, let's start from the patient, from one of us, for example. You get a drop of your blood, you spot it on the chip, you recover it, and you analyze it with MALDI, like mass spectrometry. You get the differential patterns of protein expression. And that's where the fun starts, because now you have to deal with something like this. So that you have to deal with thousands of peaks with different intensities. What do you do with that? Now you have to discriminate what is a signature of a disease from what is just a baseline. And that's where the bioinformatic tools become very important. So at the end of these, you just purify a few good leads, so a few peaks that are informative. And what you can do at that time, once you have that information, you can either predict what will be eventually the disease that patient is experiencing, but you can also do another thing. You can monitor in real time the efficiency of a treatment. So if there is one protein that you know that you're targeting and that it's a bad protein for some reason for the cell, and you have to see that protein level, the signal coming from that protein go down during the treatment. If that doesn't happen, you're not doing the right treatment for that patient. And that's how we envision this personalized medicine approach with this chip. I mean, this should really guide the clinician in identifying what's the right treatment for the patient. And this is the second, that's the second technology that we developed in our lab, and it's everything on the prediction fails, you still have to treat the patient, and that's our approach to the targeted delivery. And I added this new word that is directed delivery, because so far targeting has always posed a big problem and has never really worked that well. And so we tried a different way. And by directed, what I mean is that we want to direct the particle that we inject in the body to the right side just by playing with the geometry, with the physical aspect of the particle. And there are a lot of things that you can do. In my lab, one of the things that I discovered when I moved in this lab is that I was used to a very typical biology lab with a lot of biologists and biochemists. This lab is like a scientific orgy. You have mathematicians, physicists, engineers, chemists, and everybody can really add quite a lot to the definition of the problem and to the solution of the problem. So, of course, the holy grail of drug delivery is to develop something that can prevent the uniform distribution of a drug, like this compound, like a chemotherapeutic throughout the body. The ideal drug should recognize the site, deliver the drug, image the localization, and maybe even report the outcome. So this is something that is ideal. Now, there is a lot that you can do to address some of the issues that a drug or a particle that is injected in the body has. And these issues are mainly called biological barriers. So it's something that we never consider. We always say, okay, targeting, but before getting to the cell membrane of that cancer cell, that particle has to really start a voyage, a trip inside your vessels that is dangerous and long and full of obstacles. There are the hemorrheology, so the dynamics of the vessel, of the blood flow are an issue. The reticuloendothelial system that try to remove everything that we inject into the blood system is an issue. You have the endothelial barrier, you have the tumor-associated osmotic, oncotic pressure, you have interstitial pressure. You really have to fight against all of these to finally get to where you want. And what we envisioned was that basically it's asking too much to a single drug or to a single particle to do all of this, to tackle all the different biological barriers and to solve all of them. And so what we did, we proposed this multi-stage delivery approach, in which we basically designed the particles to do exactly what they should do, that is to get closer to the vessel wall where all the targets are. I skipped just for brevity this, but basically our mathematical models predict that if you have like a spherical particle the size of a liposome, 200 nanometers, those particles, because of the hemorrheology, they will just stay in the middle of the bloodstream, and you don't need them. They don't do any good. So the reason why we started to engineer our particles was that now you can really make them in a size and shape that can allow you to predict the way they will behave inside a blood vessel. And this is the approach that we propose, and people really liked it. And we got even the cover of Nature Nanotechnology. That was a great honor for us. And the concept is that we want to beat all these bio barriers that prevent the delivery of a drug to the right side by using a multi-stage approach. So the higher number of stages you can use, the better. And each stage has to just have to deal with only one problem and one barrier at a time. Otherwise, it's not going to work. And so basically, this is our approach. And our particles arrive. It has the right antibodies for targeting. It recognizes the right thing. It releases the second stage that is contained inside. It destroys the cancer, eventually leaves the body because it's made of silicon and it's biodegradable, completely benign. Okay, these are our particles. You see, we have been working on the design for quite a lot. They have this concave hemispherical shape that gives them particular properties. They have these pores that go all the way through, I'm sorry. It's impossible to see this. Yeah. Now, the pores go all the way through the cross-section. So, and we can tailor the size of the pores. So the pores can go from 6 nanometers up to 140 nanometers. So with this approach, we can basically load any type of nanoparticles I've heard about in this meeting. And this is one of the things that we like. We really wanted to build an approach that could serve the community of scientists and maybe help them deliver their nanoparticles to the right side. What we've done with these particles, we've tried to load quantum dots, carbon nanotubes, nanoliposomes, micelles, gold, iron oxide, chitosan, you name it, we probably already tried to load it. And we have already tried to do some study of where the particles, the second stage nanoparticles load, and you see there are different areas of accumulation. This is just a 3D rendering, but these are actual images at the confocal microscope. And we have used TEM and cryo-TEM and tomography to see how deep these are gold nanoparticles, how deep they go inside the pores. If you have very small pores, they don't even get inside. They're stuck all on the surface. If you have bigger pores, they get inside. If you have giant pores, they get all the way inside them, and you can quantify how much they load and so on and so forth. You can, whatever, I can skip this. Now, of course, if you want to build a delivery system, you need 3 things. The system has to be loaded, the system has to release, and the system has to deliver. So I've already showed you the loading. This is the release. The release is sustained over time, can be tailored, can be controlled, can be triggered by pH, by enzymatic release, by UV. There are a lot. We develop all type of chemistry on these particles. And the good thing is that it's sustained over time up to 20 hours. That's something that you want. And the final step, they deliver to the right cells. These are UVEC cells, and these red big spots are the particles loaded with carbon nanotubes and quantum dots. And you see that small vesicles are the carbon nanotubes and quantum dots that have been internalized. Now, how do these particles interact with cells? That's something that you really want to know when you inject something in the body. And apparently, the cells really like these particles, because in 15 minutes, we've done the studies. They start to project the pieces of membranes. It's like they're hugging these particles. They're very eager to internalize them. And we found particles with cells with up to 20 different particles inside. And so they're completely benign. Again, they're not toxic. I can skip this. I don't have much time. That's another very interesting thing. We don't know how these particles escape from vesicles. They get internalized. They are inside vesicles at very early time points. You can see that there is like a membrane around that's TM, nothing inside. Then you wait a few hours, and that's what you see. I mean, inside the particles, you see cytoplasm. So somehow the particles escape from the membrane that contain them inside the lysosomes, and they got it to the cytoplasm. We really don't know how they do that, but that's for sure that's something very beneficial in terms of drug delivery. Okay, the particles degrades over time. Another interesting thing is that you can control the degradation by tailoring the porosity, the pore size, and so on and so forth. The chemistry, the way they degrade, it's different according with the geometry of the particles. So, for example, if you have 2 different type of nanoparticles loaded in the external corona or the inside in the inner ring, you will have 2 differential release time. The first one to go will be the one in the outside corona. The second one will be the one inside the ring. They are completely, the cells, they're not toxic for the cells. And actually, I want to show you these movies, because this is something that we noticed, and I really like this, because it made me think if the cells can count, because we feed these particles to cells, and the cells they internalize a lot of them. And then when they divide, it looks like they evenly divide the particles between the two daughter cells. And so now I have one of my PhD that is trying to understand if cells can count, and I think it's kind of cool. Of course, we have injected them in animals. We've done all the biodistribution. They localize in the tumor much better if you add a targeting agent like VEGF receptor 2 antibody. We're doing now some near infrared with gold loaded particles. We've done MRI, and I just want to skip all of this. I just want to thank my wonderful team, the Nano team. And again, as I said before, we have people that does mathematical modeling, people that do engineering. We have a few postdocs that do chemical modification, and of course, the biomedical application is another big part of the team. And it looks like we're all having a lot of fun doing this, even if we work a lot. Thank you.
Thank you.
Thank you.
For that. Are there some questions? Question right here. Please tell us your name and affiliation.
Jean-Pierre Leberton, University of Illinois. I just have a simple question. How well can you control, I know you mentioned you didn't actually make the pore, but can you tell us a little bit about the uniformity of this pore, meaning the shape?
You're talking about the first technology or the second? Because they're completely different.
The second technology.
So okay.
Yeah.
In the second technology, that it's a top-down. So you basically have the wafer, the silicon, the bulk silicon wafer, and you etch it out just through electrochemical etching. So you create pores, but you cannot very well control the structure of these pores. You can control the size, but of course it's always a distribution of...
Yeah, but when you're talking about the diameter of the pore, what do you mean by that if they're not uniform? Are they cylindrical pore?
No.
Well, so when you say the diameter, this is a narrow aperture of the...
Well, it's the BJIH measurement. So it gives you like the distribution of pore sizes. It gives you the area, actually. And then you can approximate as it's a circular pore, but it gives you the area. So the original data gives you the area of the pores. But we can very well control the range. So we have 6 nanometers, 10, 15, 20, 30. We can tailor that pretty well.
Yeah, Rob Burgess. Yeah, Rob Burgess, Med Nanotech. Ennio, when you loaded your single-wall carbon nanotubes into the pores, did you have to do any kind of a size selection or a cutting of the tubes to get them to fit
Perfect
properly?
question. Those were like ultra short. They were like 30, 40 nanometers.
Okay.
They were pegylated, and they were 30, 40 nanometers. They were cut. Otherwise, we would have never got the long ones. We tried. We couldn't get those inside.
Your first presentation on the distinction of multiple proteins is very reminiscent of work that was done going back to 1950s, with the development of gel electrophoresis. And then there's a large literature on the proteomics of the polymorphism trace as we looked at the proteins before you can look at the genome. Now, I imagine this technique will give you even further information for individualization, that is looking to the different.
Yes.
Has there been a great deal of work on that is looking for the different alleles based on the protein?
Yeah, we haven't done that. Now, what if I got it right? You're talking about understanding the different polymorphism
Yes.
of a person on the protein the protein level.
Yes.
That's something that I've discussed with Mauro many times, with my boss, and
We ought to talk about that because there's a large literature on that.
it's not very interesting because it really is focused on cancer, and so but we've talked about that. And there is we can do the phosphoproteome
Yes,
because
I'd love to.
there are a lot of, according to what chemical group you put on the
Right.
surface, you can select like glycoproteins, phosphoproteins, and so on and so forth. So there is a lot of information there.
You can do this quickly, can't you?
It takes 10 minutes.
You could, just for forensic analysis, you could have on crime scene.
We're talking now
analysis,
with the Department of Defense, and they're very interested in getting those.
We have another question.
A similar type question, but in terms of the information you get from proteomics, number of labs are now asking the question, it's more than just proteins, that with mass spec and/or NMR, understanding the lipidomics and the metabolomics and the microRNA environment of a tumor cell in context with the proteomics is truly the way to define what is a cancer cell, or more importantly, what is a cancer stem cell. So certainly this technology is incredible, but certainly it should be, start thinking about how we can combine it with lipidomics and/or metabolomics.
I completely agree with you. And the reason why we decided to develop this technology, though, is that there are a lot of approaches that are already very good on metabolomics and genomics. Proteomics in the past 10 years, and especially like serum proteomics, got to a halt because of a lot of artifacts, and they got even advertised, and people got very skeptical about it. And so when we decided to start developing this technology, we decided to start exactly from the issues that blocked the previous technologies to get to the clinic. There were reproducibility, sample treatment and storage, prevention of degradation. And so we now believe, and we've been working on this for 3 years, without saying anything, we've been working and working. And now we feel kind of free to say that it's a safe technology, doesn't give you any artifacts. The proteins don't degrade. We were truly amazed. But of course, I mean, just by looking at proteins, you will never get the total feeling of what's happening in that tissue. Also, because it's very important also what is happening in the surrounding tissues, not only in the tumor. So I completely agree with you. But we don't have an integrated approach. That keeps us quite busy already.
But that's where telemedicine, I mean, the whole idea.
Well, very exciting possibilities.
Thank you.
And thank you for that
Thank you.
excellent talk.